In this video, we are going to talk about some information on writing your materials and methods section of your research project. I'm going to talk about some of the subsections as well as some information that will go within those subsections in your paper, and I'm going to give you a practical example as well. You'll be writing this section first for your project, and generally it's considered the easiest part to write. The materials and methods section is a critical part of the manuscript because all science should be reproducible or repeated, and this is done to be able to test if your results are actually valid. So when you write this section, you need to communicate your project in a way that others can follow and be able to easily repeat. Therefore, when you write this project, you will need to communicate in a way that others can follow and can easily repeat what you did. You need to be direct, and clarity is vital. The use of flourishings in your writing should be avoided as much as you can. Now, some common verbs that you will use to write this section are investigated, evaluated, and performed. And it is okay again to use third person. You might say, we investigated, I evaluated, I performed. For this course, you will likely have 3 to 4 sections in your materials and methods, and some parts of these are a bit flexible. The first section would normally be called something like animals, management, or procedures. When I write, these are typically grouped into one. So that's how I'm going to discuss them in this video. But if you want to in your paper, it is okay if you put them in separate subsections. Next is the experimental design. And the experimental design also can be called other things such as experimental regimen or the study design. And the third portion in the materials and methods that we're going to talk about is the statistical analysis section. Or for this class, it may simply be some calculations. Okay, so in subsection 1, the animals, management, and procedures, I'm going to break those down into each of those. So animal information would include the number of animals used, the weight, and this is specifically if it's relevant. If you had a group of mice that were all more or less the same, the weight may not be necessary, but if you were looking at beef cows and you wanted to track their growth over time, you would want to report the starting weight. You would include their age, especially their breed, because some results could be breed specific. You would want to look at their sex classifications. And as you remember from earlier in this course, the sex classifications can include whether or not, say, they're heifers, bulls, they're boars, etc. You would also want to report their parity. And this is simply the number of times that a dam has given birth. Now we move on to the management system. You're basically reporting how you controlled the research environment. This includes their diet and feedstuff used. It includes their housing or their, say, caging system. This also will include the dates and location of your experiment, because this gives some indication of the type of environment these animals would have been reared in. It's very different to do a research project on pastured animals in the winter versus in the summer. Finally, you also want to be sure to indicate any handling techniques that are relevant. For example, my cows, when I do research, are all on pasture, and we feed them individually different types of selenium. And to do this, we have to use a special device called a kaolin gate. This is where when a cow comes up to a feed bunk, they're wearing a collar that has an indicator, and when it comes up to its specific bunk, the gate will open, and only for that one cow. This is something we have to always report that we are using in our papers because it affects the way that we are looking at the animals or the model that we're using to analyze the data. And next here, we would have the procedures. And this is simply indicating your treatment groups or what you're studying. And this is typically going to be introduced in the introduction or objectives. For example, say we're studying 2 levels of calcium and the effects on growth. You would want to highlight, say, diet 1 had a certain level of calcium, and then diet 2 had a different level of calcium, and you want to report these levels. This is a great place where you could use some visuals. 2 visuals that would be very handy in this situation would be either a figure of what's happening, a layout of the management or the environment, or a table. An example for a figure would be, say we wanted to look at the effects of introducing a rabbit from a very calm environment, and we took it and we placed it in a very stimulating, overly stimulating environment, and we wanted to see how the animal behaves. Well, we might use a figure to describe the 2 different environments. Say in cage 1, it's the easy environment. They have their feed bunk. If they're using litter boxes, they have the litter box. And then they have the water. And it's very simple. But in a stimulating environment, you might have the same, the litter box, the water, but you may have added, say, some moving parts on this side, some things hanging from the ceiling to try and stimulate or get a reaction from that animal. And so you could use a figure to display what is happening. Another thing you can do is use a table. And this is commonly done if you're reporting different diets that you're using or nutritional information from a diet. And to do this, you could put treatment groups, say low calcium or high calcium from that first example, and then you would list your nutrients on the side, calcium being the most important, and list the contents. Most of the time, if you're studying, say, calcium, the calcium is the only one that's going to differ, but the rest would stay the same, which is important for a study. But occasionally, if you affect crude protein, you might also affect TDN, and so it's important to show those 2 different diets. Finally, in this first major section, there's typically a statement about approval from an ethics committee, and you'll have an approval number for a protocol. This is not relevant to this course, but when you're reading research papers, you will most likely see this statement. The next major section is your experimental design section. This section basically just says how the experiment was conducted. You will state specifically what was measured and how you measured it. And you can even state, or even right here, when you measured it. And this is another good place that you can use a visual. I personally like to use timelines in my experimental design section because they show kind of a stepwise of what is happening. The main part of your experiment may be time point 0. This is when your experiment had started. And then say you're feeding different levels of calcium. You feed it for, 1 week, 2 weeks, 3 weeks. And then on each of these days, you take their weight. This is very simple. And so timelines are really easy for the audience to follow and get a snapshot idea of how you set up your experiment. finally, there's going to be a statistics section. You will have a video to watch on working with data and how to report data. And so I'm not going to talk much about statistics right now, but you'll be directed to that video when it's time to write your results section. There's going to be many differences in terms of how the data and the statistics is reported. For some of you in the class, you may be able to use a simple formal test such as a t-test, but most of you are probably going to have simple calculations of just calculating the means, which is the averages of your responses. Right, so that's the basic outline. Please use this outline when you are writing your materials and methods for this course. And now that we've gone over it, I'm going to show an example. Okay, so this is a paper I recently published. It's a very simple, clear, straightforward paper, so it's a good one to actually use for this course. On this top, you can see at the top here just lists materials and methods, and then I have subsections. And even some of these subsections have smaller sections, but for this paper, we're not going to go that far. There's the materials and methods, and then there's subsection on animals and the experimental procedure, the experimental regimen, which in this case, there were 3 different mini experiments within this paper, so it's a little bit more complex in the course. I have a section on the selenium and hormone analysis, and then the statistical analysis. So it's very basic, very similar to what we just talked about. The first section, and I have it written over here on the right, is the animals and experimental procedure. So there's the approval of protocols, which I told you existed, but is not relevant to this course. So for this paper, is written first. So this is here in blue, where's the approval of protocols. And then we have the animals, breed, and age. So I'll make that a different color. So we have animals, breed, age, 11. And this also includes the parity. As you can see, multiparous means they've all had more than one calf. We have diet and management can sometimes be sort of the same. So we're gonna go with diet first. So we have diet here. The forage based. But then we're gonna go back and do management. So all of this is talking about management. They're managed in a fall calving production system. And how? So this is the location of where the cows were. So location of experiment. And then we go back. This is more diet. This is how we fed our cows. And then I go into the setup of experiment. Let's make a completely new color for this. Light colors. Let's go with shiny blue. The setup of our experiment is we chose our cows from specific cow herds that we've already been using. And then our treatments, which is a very, very, very important part of this study, is we have our treatments. These are dietary treatments. So again, it kind of even correlates again. We have our treatments and very specific about what is happening here. And so that's a breakdown some of the materials, the animals and experimental procedure, containing the different parts that we just talked about. In this course, each student will have a similar section. It may not sound quite as technical as this, but there will be some sort of section that describes this information. Next, there's the experimental regimen. Remember, this is how it's experimented, how it was conducted. For this case, we did transrectal ultrasonography, and we talked about our synchronization methods. We discussed what we monitored or measured and how we measured these. And so, let's see. The major part of this is where it starts the luteal function. It was converted ultrasound. And then it's our synchronization protocol. And we're very specific. In research, we have to write the drugs and where we get them. For this class, it's not as necessary. Then monitored for estrus. Then what we did, what and when we measured them, and how we did that. And finally, what we did with those measurements. So that's the important part actually the sentence covers all of these. If I was going to take this and include a figure, my figure would probably be a timeline. I'm a big fan of timelines again. See, we draw out the line, and what we did, day 0 was considered experimental days, the start of experiment, in which estrus. So prior to that, we gave PGF2 alpha to synchronize estrus. Estrus is day 0, and then on days 4, 7, and 10, we collected blood to test for progesterone. That might have got a little complicated. If you have questions, feel free to email me, text me, catch me in class. All students in this course for your paper will have a section similar to the experimental regimen or experimental design. In this paper, we had something called the selenium and hormone analyses. This will not be required for our course. This gets very technical and very detailed. This is how we took our blood samples. We in the lab, it talks about the machines we used. It talks about how every step of how we analyze the amount of progesterone in those samples, and it's way too much for this course. But I did want to show that oftentimes it is present and quite required in terms of being in the materials and methods because it is vital for the reproducibility aspect that we talked about previously. Right, finally, there's the statistical analysis. We don't have to talk much about it here, but in this case, it basically says there was an ANOVA using a computer software and a significance level of 0.05, and that's basically it. For this course, that's way more than you're going to have to say. There may be a t-test using Excel Max. You're not going to use any sophisticated software. You'll be reporting the means, which is simply averages. And we will probably still use a p-value of 0.05 for those students who are actually able to generate a p-value. That is all I have for you on the materials and methods. If you have questions, reach out anytime.